techniques and cell culture. We'll explain the purpose of aseptic techniques, explain how microbial culture can be started, suggest which chemicals might be added to media to allow the growth of animal cells. We looked at why liquid cultures are often plated out, and then the technique required before plating out concentrated cultures to allow colony counting to work. So aseptic techniques involve sterilizing the equipment and culture media by the use of heat or chemical means and the subsequent exclusion of microbial contaminants. It's very important if we are taking bacteria from a stock or a culture that we don't contaminate our stock solution so that the equipment we're using is clean. And also that when we're transferring our microbes, we only transfer the microbes that we want, not the other ones that might be growing on our equipment. So as I stated before, we can use heat or we can use chemical means like Virkon and alcohol solutions. Many culture media exist. When we're growing a microbial culture, we can start by using an inoculum of microbial cells on agar. So we might already have this situation here, several cells growing out in different places. We might say we want to take this cell and use it to start a new culture. So we would take our flamed inoculum loop, we'd lift this off and move it somewhere else. So we can use microbial cells on agar, or we can use a broth with suitable nutrients as well. So there's lots of different ways we can do this. There's many different culture media that can be bought in or made up, and each will promote the growth of specific types of cells or microbes, depending on which growth factors have been included and which nutrients have been included. Growth factors are proteins that promote cell growth and proliferation. They're essential for the culture of most animal cells. So just taking a cell from the human body, for example, and putting it on agar will not work. We need to know what we're going to need for the animal cells to grow and how to grow it. And again, we're going to need to use aseptic techniques. Plating out a liquid microbial culture on a solid medium will allow us to count the number of colony forming units that we have. So if we take a liquid culture, say 1 mil, and spread it out across a agar plate, we'd then be able to count how many different colonies we had transplanted onto the plate. We previously discussed in our log dilution video that sometimes we need to dilute the samples out. So for example, if your 1 mil solution has 1 million colonies on it, there's no way you're going to, when those colonies grow out, be able to count them on a 10 centimeter dish. So we have to dilute them down. So for example, if you diluted it down 100,000 times and then found 10 colonies, you're looking at 1 million colonies in the original culture. So we multiply the number of colonies found in diluted culture by the dilution factor. That allows us to estimate the original number of cells in the stock, which is known as the colony forming units, or CFU. This will tell us roughly how many cells are present per unit volume, which is known as the density. So if we know there are 1 million cells in 1 mil, that would be the number of cells per unit volume. So as we said, serial dilution is very important, and in this top left plate, that might be the completely undiluted culture, which obviously gives us the most accurate count, but there's no way you're going to be able to count all those cells. So if we look across, all of the plates on the top are fairly useless for counting. I would say the one at the bottom left is probably the most useful. So our concentrated cell cultures have given confluent growth where the cells are growing side to side and they can be easily counted. We would use serial dilution. It's important not to dilute too much. For example, this sixth plate at the bottom right only has 2 colonies. If we'd included an extra colony, that would increase the count by 50%. So if that bottom right one is diluted down to 1 millionth the original colony concentration, then an additional cell would add 1 million colonies to our count. Another way we can do our count is through hemocytometry. So hemocytometers can be used to estimate cell numbers in a liquid culture. A very small volume of culture is placed into this internal well, and the number of cells in that area are counted, and then this number is scaled up again to the volume of the original solution. So if we had a litre of the original solution and we put 1,000th of a mil into this plate, if there are 5 cells, we can multiply that up. So one issue in microscopy is that dead and living cells can appear identical when viewed. Vital staining is the technique we use to count the number of viable living cells. One type of vital staining shown here called Jurkat stain is actively excluded from living cells. So when it diffuses into the cell, if they're alive, will be able to push it out. That means we can count those that are clear as the living cells, and we can see here that are cells that have not been able to kind of get rid of excrete the stain. So those ones are probably dead. In this video, we have looked at aseptic techniques and cell culture. So aseptic techniques involve the sterilization of equipment and culture media by heat or chemical means and the subsequent exclusion of microbial contaminants. A microbial culture can be started using an inoculum of microbial cells on an agar medium or in a broth with suitable nutrients. Many culture media exist that promote the growth of specific types of cells and microbes. Growth factors are proteins that promote cell growth and proliferation. Growth factors are essential for the culture of most animal cells. When plating out liquid microbial culture on solid media, it allows the number of colony-forming units to be counted and the density of cells in the culture can be estimated. Serial dilution is often needed to achieve a suitable colony count. We've looked at the method and use of hemacytometers to estimate the number of cells in a liquid culture. And we've looked at vital staining and why it's required to identify and count viable cells.